Warburg effect. In the healthy tissue, the blood vessels provide nutrients and oxygens. When tumor grows faster than its blood supply, the blood vessels are poorly organized. As a result, the inside of the tumor usually is a low oxygen environment, so-called hypoxic environment. Healthy tissue focuses on ATP production and homeostasis. Cancers, in contrast, focuses on proliferation and survival. To achieve the goal, cancer cells change the metabolic pathways. They increase glucose uptake, and by changing the metabolic pathways, they produce more lactate rather than produce ATP. This kind of metabolic changes we call Warburg effect. In normal cell metabolism, glucose enters glycolysis pathway and it generates NADH and ATP. And then the glucose is divided into two pyruvate motif, and then pyruvate is changed into acetyl-CoA. This acetyl-CoA enters the TCA cycle, and as a result, NADH and FADH2 is formed. And through electron transfer pathways, this NADH and FADH2 is oxidized by oxygen, and it generates the high number of ATP and water. In cancer, this pathway is changed. The pyruvate, rather than go into the TCA cycle, through fermentation process, it is changed into lactate, and all other downstreams is blocked. This process is energetically less efficient, but faster in rate. And the formed lactate is being used for building blocks for cancer growth, and also the lactate can be regulator of tumor environment. The oxidative phosphorylation in mitochondria is reduced. Instead, maybe the ROS stress can be regulated. If Warburg effect is selective only in cancer, maybe this change the metabolism can be good target for new cancer drug development. To summarize, cancer cells has a high rate of glucose uptake, also fast glycolysis pathway, and unique fermentation pathways from pyruvate to lactate. And the enzymes involved in glycolysis is overexpressed, and hexokinase 2 is one of them. And lactate dehydrogenase A, which is involved in fermentation, is also overexpressed. HIF, hypoxia-inducible factor, is the controlling molecule which can overexpress these proteins. So, glucose transporter inhibitor can block the glucose uptake, and hexokinase 2 inhibitor can block or slow down the glycolysis pathways, and lactate dehydrogenase A inhibitor can block the fermentation process. These inhibitors potentially can be selective only for cancer and may have low side effects.